This is the first lecture for Monday, April 13th. So we're continuing on with our survey of different marine ecosystems, and we're moving well offshore from the coastal ecosystems, and we're moving to these specialized marine environments, harsh marine environments, where there's some kind of physical challenge for the success of marine organisms. There's adaptations that you see in marine organisms to deal with these environmental extremes. So we're going to look at two specialized marine ecosystems. The deep sea, which is very extensive, and then polar ecosystems, which are also pretty extensive. So to begin with, I wanted to comment on this Dropbox assignment that you guys had the other day, just to inform you what the results were. The question that you were asked was, what is the most highly adapted vertebrate group in the ocean? And these are the results. The number one choice was fish, followed by cetaceans, sharks, penguins. Then you can see there were a number of other ones. So there's not that many vertebrate groups besides fish. There's thousands of species of fish, not so many species of these other things. But one of the reasons why they're so interesting is because of the adaptations that they have. Even if there's not very many of them, they're still quite interesting. One of them, the sea otter itself, well, we had an entire lecture on the sea otter. They certainly have some interesting adaptations for life in the ocean, very different than some other marine mammals. But back to the deep sea. We've looked at these different marine ecosystems, starting with the ones that are close to shore, and there were a number that we looked at. We looked at this kind and that kind, this marine ecosystem, that marine ecosystem. So there's a lot of diversity close to shore. But in terms of how extensive a part of the ocean they are, some of them obviously not very extensive. Some of them very productive, some of them that humans have a lot of interest in, but others a lot more extensive. And this is an example, is the deep sea. So if you look at the average depth of the ocean, it's about 4,000 meters, the average depth. All of these different ecosystems that we've looked at so far have been in relatively shallow water. Even when we're looking at the continental shelf ecosystems, that extended out to a couple hundred meters, not thousands of meters, but a couple hundred meters. And maybe 100 miles, 200 miles from shore when you have a continental shelf that extends a long ways from the continent. This is a huge area of the ocean. You can see in this slide that it says 75% of the ocean reaches depths greater than 3,000 meters. And you may remember that the Marianas Trench, the deepest part of the ocean, is about 11,000 meters. So if you go out away from the continents, if you look at the figure down there, there's the continental shelf and there's a continental slope, and you get down to this area where there's a different plate of the Earth's crust that's not continental. The continental plates are basically floating on top of the other parts of the Earth's crust, moving around in that continental drift, that plate tectonics. So there's these vast stretches of abyssal plains. There's ridges, mountain ranges. There's a lot of topography there in the deep sea. And it's a specialized environment in a number of different ways. So there's a lot of pressure. There's not very much food. It's cold. and there's no light other than light that's derived from biological sources. All these different places that are close to shore are relatively easy to study. Thousands of years people have been gathering information about what you could collect when you're walking along the shore. You could wander down to the tide pools. You could walk along the rocky shore, sandy beach. Those type of environments are easy to study. Even the coral reefs, very nice place to go. It's warm and welcoming, and there's some really nice places where there's coral reefs, and a lot of people interested in studying those. Obviously, the deep sea, you can't just get in a boat and go out in the deep sea. You can't just walk there, go there on a field trip for a class. It's very, very hard to study, very difficult to access compared to most of these other ecosystems that we've looked at. Certainly the open ocean has some similar challenges in that you have to go a ways away, long ways. You have to be in a boat. A lot of the open ocean research is done by satellite these days, but you can imagine why there's so little information that's available about the deep sea compared to the rest of the ocean, because it's a very challenging ecosystem or series of ecosystems to study. Well, let's look at what the deep sea is exactly. There is no exact definition of the deep sea, but let's put this in context of what we've looked at so far. Some of the things. Here's this old slide, but it's still very effective, this old figure that shows the percentage of the Earth's surface. So this is just the flat horizontal surface of the Earth that is a certain elevation above that blue dashed line and a certain depth below that blue dashed line. So if you look at in figure A, kilometers, the percentage of the Earth surface that underneath that blue dashed line, that's the depth of the ocean. So you look at that, 2,000 meters, 4,000 meters, 6,000 meters, there's 23.2% of the Earth's surface below the ocean, has water that's over 4,000 meters deep. Now that's pretty deep. And if you look at that curve on the right in the figure, you see continental shelf, it's a tiny little fraction of the Earth's surface. It's continental shelf, continental slope a little more, but look at this vast extent of the deep sea. There's a very large percent of the Earth's surface that if you were on the surface of the ocean, you went down, that you'd be going into the deep sea. You'd be going into water that's very deep. So the point is that however you look at it, it's a very, very large part of the ocean. So let's look at the conditions in the deep sea. And there's a number of different conditions, but in terms of being an extreme environment, in terms of having environmental conditions that organisms have to deal with to be able to survive there, there's 4 that we're going to look at. Very cold, very high pressure, dark, and low biomass. So we're going to spend some time looking at those conditions, first of all, and what kind of adaptations marine organisms have in response to those. So this is basically what the deep sea looks like. If you took out those yellow letters and you made yourself very cold, this is what the deep sea looks like. So that's what you'd experience there. There's no light other than what's coming from animals. It's cold, it's dark, and it's high pressure, and there's not very much food there. So let's look at some of those conditions. Let's start with high pressure, and let's look at cold. those go along with each other. As you go down in the water column, then obviously the pressure increases quite a bit, but it also gets very cold very quickly. So if you talk about these depths, those of you that are scuba divers, you're familiar with this idea that you go down 10 meters, or about 30 feet, 33 feet, and the atmospheric pressure doubles. You go down another 10 meters. So you're down at 66 feet or so, then there's the atmosphere doubles again. It doesn't double again, but it increases by another atmosphere of pressure. So just down at 66 feet, then you're experiencing pressure that's much greater than at the surface. And 66 feet, as we saw, is not what the typical depth of the ocean is. It's thousands of meters, 3,000 meters average depth. So if you look at this curve over there, just in terms of pressure, you'd see a really dramatic increase in pressure as you go deeper. But the other thing is that it gets very cold. And we looked at the thermocline, this characteristic water where the water temperature drops very quickly. It creates layers, boundaries in the ocean. There's a lot of things that don't move down very much past the thermocline. But the point here is if you look at the water temperature, if you look at the depth, it doesn't have the depth gradient on there, but other than the 3,000 meters. But if you look at that figure over there, you can see that it gets deep pretty quickly, and then there's a huge part of the ocean below a couple hundred meters where it's 5 degrees or less. That's really cold. 4 degrees, 3 degrees. It's really cold. So the upper part of the ocean, the upper layer of water in the ocean, it can be relatively warm, but you don't have to go very deep to get into cold water. And you certainly don't have to go very deep to get into high pressure water. You go down a couple hundred meters, it's cold and it's high pressure already. So those are two obviously kind of extreme environmental conditions that you have to deal with in terms of being an organism that lives in the deep sea. So what kind of adaptations do organisms have in the deep sea? Well, in response to cold and in response to high pressure, one of the things that's very different between shallow water organisms and deep water organisms, even very closely related in the same fish or crustaceans in the same genus, just a different species, but they're very similar. One of the differences is the enzymes that they have. They have the same enzymes, but they have different versions of these enzymes. They have enzymes that function optimally at different temperatures. So that's how enzymes work. They catalyze reactions. And they have some kind of optimal rate of catalyzing a reaction at a certain temperature. And below that temperature, above that temperature, then the efficiency of some reaction that's catalyzed by enzymes decreases. And so for warm-bodied organisms, that's a very narrow range, the body temperature of that organism basically. And above that, enzymatic reactions don't occur optimally. Below that, they don't occur optimally. Well, in cold-blooded organisms, there's a broader range of temperatures that enzymes are functioning at. But if you compare the enzyme efficiency, the rate at which a reaction is taking place, and a reaction that's catalyzed by an enzyme, and you look at these two enzymes, maybe it's the same enzyme, and you look at a deep sea animal and you look at a shallow water animal, then in that figure you can see the rate of the reaction. It's optimal. It occurs the fastest as catalyzed by this enzyme in the deep sea organism at about 3 degrees. In the shallow water organism, that takes place about 15 degrees. Same enzyme, a lot of cases. So the problem with trying to keep these animals in captivity or trying to study them, one of the problems is that their enzymes get all out of whack when they're raised to the surface and experience these warmer temperatures. One of the things that has to be taken into consideration in studying them or trying to keep them in captivity is you have to keep the water very, very cold, or their enzymes aren't going to work. If their enzymes don't work, then that's trouble, and these organisms can't function. They die a lot of times. So this has developed over thousands of years, millions of years. Their enzymes, through natural selection, have come to function optimally at these low temperatures. You'd see the same thing with pressure. Pressure affects the enzyme function, enzyme kinematics in a similar way. Now, you may remember back from your biology days that every cell membrane, or every cell has a membrane, and this membrane is primarily made up of phospholipids, these special type of lipids that have a charge and they orient a certain way. Well, the cell membrane is made up of this lipid bilayer with a glycerol molecule and these phospholipids that come off of these fatty acid chains. And this is different than triglycerides, which is the typical type of lipid in animals. But there's phospholipids that make up the cell membrane. If you look at this figure down in the left, that blue figure, where there's water on the inside of a cell, there's water on the outside of a cell, and there's a cell membrane that's separating the inside from the outside of the cell. Well, those things in there are phospholipids. So instead of having a glycerol with 3 fatty acid chains, phospholipids have these 2 fatty acid chains that stick down there. Those are those red things that look like tails, those 2 chains of fatty acids. Well, if you look at the composition of those fatty acids, those are carbons that are joined together. And in shallow water animals, most of those carbons are joined together in single bonds. And if you remember anything about chemistry and bonds, you know that between some elements there's single bonds, and between others there's double bonds. There's the existence of double bonds. And so if you look at that saturated fatty acid in figure A, where it says shallow water animal in the blue, you notice all these carbons. There's a glycerol molecule there at the top, but all these carbons have a single bond between them. There's a CH2 and a CH2 and a CH2. They're all single bonds, one line. This palmitic acid, that's a typical fatty acid in a shallow water animal. No double bonds. If you look at an unsaturated fatty acid, B, in that figure, then you see those red arrows are pointing to these double bonds. There's double bonds between the carbons that highlighted in yellow. That's what you tend to find in a deep sea animal, is you find a lot more fatty acids in their phospholipids that have double bonds. And so instead of being this long, thin chain of carbons, or fatty acid chain that's highly organized, you have these ones that are kinky. Kinky in the sense that they contain double bonds. And so what that does is it keeps the phospholipids, or at least the fatty acid tails of these phospholipids, from being packed together tightly. And what that results in is that the phospholipids in the cell membranes of deep sea organisms maintain their fluidity. So they're moving around in the cell membrane rather than being packed together, which would decrease their fluidity, and it affects the transport of substances across the cell membrane into the cell and out of the cell. And in order for organisms to function properly, there's all kinds of transport of substances. This is something else that you learned in your biology class, that there's diffusion, there's osmosis, there's active transport, there's passive transport, there's facilitated transport, all these different mechanisms for transporting things in and out of a cell. So if you look at those carbons in the fatty acid tails of phospholipids, you see a lot more double bonds in deep sea organisms than in shallow water organisms. This is true of fish, it's true of crustaceans, it's true of squid, mollusks. And so, as I said, what that does is prevents them from being packed together very tightly. And what that does is it maintains the fluidity of the cell membrane and maintains the ability of the cell to transport substances in and out of the cell, which is an important characteristic for living things. All living things have cells, and those cells have to move things in and out of those across the membrane. Now here, if you look at the percentage, this is a figure that shows the percentage of unsaturation, or it's basically a reflection of the number of double bonds that are in between the carbons in a fatty acid tail that's in phospholipids. And so here we're talking about temperature, which has a similar relation, very cold, more double bonds, because very cold water decreases the fluidity of cell membranes, same thing that high pressure does. So if you looked at pressure as opposed to temperature, you'd see a similar trend. So in this figure, there's these Antarctic species. If you live in Antarctica, it's obviously very, very cold there. If this was pressure, increase in pressure as you move to the left, you'd also see this increase in the unsaturation, the percentage of carbons that are unsaturated, or the percentage of phospholipids that have double bonds. And again, what that does is maintains fluidity of the cell membrane, which is an important adaptation for deep sea. So if somebody showed you if you had a cell and you looked at the cell membrane and you looked at the phospholipids and you looked at the number of bonds in the fatty acid tails of phospholipids, you'd be able to tell whether that organism was from the deep sea or whether it was a shallow water organism. All right, well, that's a good place to stop for now, because we can move on to some of these other adaptations in the second lecture.